Okay, here we are back again with our generated, looked over, but not yet edited Coxiella burnettii database. Now the first thing I'm going to take a look at, as I said, is how we edit and what we can do with reactions. So with that in mind, we're going to want to take a look at what's actually inside a reaction frame. And remember, this is a frame-based database, so functionally the same thing as a page, but the frame is where the information is stored. So let's go find ourselves a reaction. I'll go up to Reaction. I'm going to search by EC number. And I'm going to go for 2724. And no, I don't have this memorized. It's in my notes. Okay, so this is the aspartate kinase reaction from lysine biosynthesis. Just the conversion of aspartate to aspartyl phosphate. So the very basics of editing start with right-clicking on an object to edit that object. Okay? So if we right-click on the EC number here, which is the reaction name in this case, we get this menu asking us what we want to do here. Do we want to show the history? Do we want to show any notes, which there probably shouldn't be? Do we want to edit? And yes, we want to edit. So I'll click on the edit, and notice my many editing options. We have the reaction editor, which is where we're going to go. We also have the create/add enzyme option, and that's how we connect an enzyme to a reaction. And that will create this intermediate enzymatic reaction frame that's kind of invisible to us but is there regardless. But that's how to make the link. And you can make the link from the enzyme side or from the reaction side. This is how you do it from the reaction side. You can detach enzymes, so you can decide that you don't actually think the enzyme should be attached to that reaction anymore. Maybe it was assigned an error, maybe you assigned it an error, or maybe you need to move the reaction around. You'll see a circumstance later under which we might need to do that when I'm looking at proteins. Finally, there's the frame editor, and the frame editor lets you look at the guts of the frame. It basically brings it up in almost a plain text format, and you shouldn't need to monkey with the frame editor much, if at all. Then there's the relationships and ontology editors. These are also these sort of under the hood kind of editors that you should be touching even less often than the frame editor. And below that, just some straightforward functions: making a frame, deleting a frame. You don't really need to create frames from this editor menu either. Okay, so with that in mind, let's go click on Reaction Editor. And this brings up the Reaction Editor interface. Okay, so let's look at the features of the Reaction Editor. Starting at the top here, we have the conversion type. So this is a pull-down menu that is mostly self-evident. Is it a chemical reaction? Is it a binding reaction? That is, protein A sticking to protein B. Is it a transport reaction? And notice I just described binding reactions that way, but this is not how you represent protein complexes, so don't worry about that right now. Transport reactions, then electron transport reactions, redox half reactions. Now you have complex processes and unknown conversions. These are both black box kind of terms. A complex process is basically a multi-step black box, and an unknown conversion is a black box. So that's when you don't have a detailed analysis of what is actually going on. So we're going to stick with chemical reactions for now. You have a box where you can put in an EC number and a check mark here if it's the official EC number. We have that here because you may use EC numbers for reactions that are not strictly the EC reaction. For example, there may be some reaction that is predicated on a certain type of amine group, and you have another kind of amine group that can also operate in that kind of reaction, so you've represented it, but it's not the official EC reaction. And you may do what we do sometimes and do partial EC numbers, where you yourself categorize it. Oh, I think this is 272-ish. So, it's the right kind of high-level function, so 272 dash. We can accept that too. You don't actually have to have a full EC number, you just have to have something that sort of follows the EC number format. And this will actually, assigning that number, even a partial EC, is useful because it will put the reaction into the hierarchy in your database in the appropriate place. Below that, we have the quick entry box, and this is where you can actually just type in the full reaction equation. And that's typically how we end up doing it. And I'll show you what that means a little later. Then you have the detailed listing here that shows each participant, reactants on this side, products on the other, or vice versa, since it might be reversible. But one side of the reaction and the other side of the reaction split appropriately. And the one highlighted in blue is the one selected for editing down below. And so when you click on a specific reaction item, that compound is then placed in the editing area down below, and we'll just scroll down here and you'll see that I clicked on L-aspartate and it's in this editing area. Now if I click on ATP, it'll be ATP down here, and this is where I get to change the compound choice. Perhaps I get to add in information like what compartment it's in. This is important for transport reactions. It might be important for other kinds of reactions too. We think the space of assigning locations to reactions and compounds is not very well explored right now, but we have a lot of capabilities there. And you can change the stoichiometry here. For example, I can change the amount of aspartate in the reaction from 1 to 3. Just click away. And now when I click back up, you'll see it's changed up here. And I am now very wildly inaccurately suggesting that 3 aspartate can combine with 1 ATP to generate 1 L-aspartyl phosphate. Now that's unbalanced. And down below here, our balance checker is actually telling you how unbalanced it is. It's telling you what it's unbalanced by. The one important note here is that hydrogens are not always properly balanced across reactions because there are issues with the protonation state of certain compounds. You kind of, as a database, need to punt on that kind of thing and just get everything else correct. Now scrolling back down again, you'll notice there's a space for a summary, there's a space for citations, there's a space for a marker if it's spontaneous, if no enzyme is required. Now, we don't typically these days use summary spaces on reactions, and it would be limited to if there were something really particularly interesting about the mechanism of that reaction in general. And this is a point where I'd like to remind you that the way we represent things, reactions are the Platonic ideal form of the reaction. They're the reaction independent of the enzymes that may carry it out. And we do it this way because that way we can have each enzyme connected to each reaction by an intermediate enzymatic reaction frame, which is invisible once you're using the database, and that contains information like the Km and optimal temperature and basically how this enzyme does that reaction. And right now we're looking at curating and editing that reaction. And so this is a matter of the chemical transformations that are taking place. And you're not going to write a lot of comments in the summary about how, say, aspartate kinase carries out this reaction. That's not where you really want to store that information, because later on you may find another enzyme that does the exact same reaction, and it'll be awfully confusing if you have information about one particular enzyme in the reaction listing. And now if we go and click OK, it'll warn us again, hey, you left that reaction unbalanced. What are you doing? And we'll just hit OK here. But now, yeah, we've left an unbalanced reaction. Now, say you have made some changes and you really kind of regret those changes now. Well, you always have the option up here of reverting them. And this is a good time to talk about saving unsaved things. So here we have the option to save the database, delete a database, isn't that a scary idea? Create a new version, and revert current database. So we're going to choose to revert the current database. I'll left click on this, and the dialog window comes up saying, really? All that work you just did? It's going away? And you say yes. And it rolls back your changes, and it's going to look through the whole database really quickly to check what those were. And now here we are back again. We have our proper balance. It is indeed 1 L-aspartate. And no, I did not have to revert the whole database back to the last save point to get that. I could have just gone back into the editor, changed the number, made it correct, and it would have been good. But if you've screwed up in such a way that you're not even clear how to get back to the original state and you don't want to save stuff, the revert feature, very handy.